Hi and welcome to the session. So in this session we are going to discuss about the topic IR spectra and applications of IR spectroscopy from unit 5, that is the spectroscopic technique part IR spectroscopy. What is the IR spectra? IR spectra is a graph in which the x-axis belongs to the wave number in centimeter inverse or frequency, and y-axis belongs to the percent transmittance or absorbance. In most of the cases, x-axis will have wave number in centimeter inverse, while on y-axis there will be percent transmittance. This graph is nothing but the behavior of the molecule in which it absorbs the infrared radiation, and it will carry out the vibrational transition in a molecule. In IR spectra, there are two main regions. First region is called as a fingerprint region, and second region is called as a fundamental group frequency region. Now if we see this IR spectra, the region of IR spectra ranging from 400 centimeter inverse to 1400 centimeter inverse will be called as a fingerprint region. And this fingerprint region is very important in order to detect the compound present in the analyte, because the fingerprint region will have exact matching with the IR spectra of any sample containing same organic molecule. So it can be used to identify two samples which are having same organic molecule content. Now further, the region from 1400 centimeter inverse to 4000 centimeter inverse is called as a fundamental group frequency region. Here the peaks obtained in this region will be due to the vibrations in a molecule containing specific functional group. This is the IR spectra of benzaldehyde, and the functional group present in the benzaldehyde is nothing but the C double bond OH. In this case, spectra of benzaldehyde, the peak obtained here around 1700 centimeter inverse will indicate the presence of aldehyde group in given compound, while the peaks near 2800 centimeter inverse will show the presence of hydrogen atom connected to the C double bond O, and it will indicate the presence of aldehyde group in given compound. So simply the fingerprint region will give us the information about the molecule if we want to match the two molecules in two different sample, while the fundamental group frequency region will help us to find out the functional group present in a molecule. Now we will discuss about the applications of IR spectroscopy. First application of IR spectroscopy is the identification of functional group and the structure elucidation. So we have discussed in the previous slide, that is the IR spectrum is divided into two parts: group frequency region and fingerprint region. Group frequency region ranges from 4000 to 1400 centimeter inverse, while that of fingerprint region ranges from 1400 to 400 centimeter inverse or 1400 to 665 centimeter inverse. In group frequency region, each peak corresponds to specific functional group. For example, if IR spectrum show a broad peak in the region 3500 to 3000 to 3200, it indicates the presence of OH group of alcohol or phenol. So if we get the peak in this region, which is a broad peak, it will indicate the presence of OH in alcohol or phenol. Hence, based on the peaks observed in the functional group region, functional group present in a sample can be determined. This functional group helps to determine the possible structure of the sample. Also, peak in the fingerprint region helps to decide characteristic peak of particular bond between the two atoms based on the vibration frequency of that bond. Such peaks will also help to determine the structure of the sample. Second application is the identification of the substances. Depending on the number of atoms present in a molecule and whether the structural arrangement is linear or non-linear, large number of absorption bands are observed in the IR spectra of organic molecule. Hence, the two molecules having same IR spectra is not possible, and based on the IR spectral data, the two samples are identical or not can be decided. So if two compounds are having same IR spectra, especially the fingerprint region, then both of them must be sample of the same substances. IR spectra of two enantiomeric compounds are identical, so IR spectroscopy fails to distinguish between enantiomers. For example, an IR spectrum of benzaldehyde is observed as follows. So here just we have discussed in the first slide that about the IR spectra of the benzaldehyde. So IR spectra of the benzaldehyde will be same for any sample containing benzaldehyde. So no other compound than benzaldehyde produces same IR spectra. Third application of the IR spectroscopy is the detection of impurities. As discussed in the last application, IR spectrum of organic molecule does not matches with the other one. Now if the test sample to be determined is compared with the standard spectra of same compound, then both should show same spectral pattern, and if any additional peaks are observed in the IR spectrum, then that additional peaks suggest that they are due to certain impurities present in the compound. Fourth application is quantitative analysis. In case of quantitative analysis, baseline technique is used to determine quantity of a substance or a mixture. Here absorbance, that is log of I0 upon IT of chosen characteristic peaks of substance under analysis is compared with the same characteristic peak of the same pure sample of unknown concentration. Then this comparison will be helpful to find out quantity of analyzed substance. The method can be used to determine quantity of a substance either in pure form or in the form of mixture of two or more compounds. Fifth application is progress of the reaction. So IR spectroscopy can also be used to find out the rate of reaction. In case of chemical reaction of organic molecule, generally one functional group gets converted into another one. That means one functional group will disappear and other gets formed. If small portion of reaction mass is analyzed by IR spectrophotometer, then progress of the reaction can be determined. Just for example, if C double bond O is converted to OH, that means there is a reduction reaction in which C double bond O is converted into COH. Then sharp peak of C double bond O, which is around 1700 centimeter inverse, will disappear and a broad peak of OH will appear. So these are the applications of IR spectrophotometer. Thank you.